Hello, everyone. Thank you for attending the presentation today. I really appreciate your time. My name is Anthony. I work with TreeFrog Therapeutics. I handle the business development activities for the North American market, and I'm based in Boston. So let's dive into the presentation. So TreeFrog Therapeutics is an iPSC cell-derived cell therapy company. And this is the three core added values of our technology. And I just give it as a snapshot early in the presentation because you will see this throughout the slides. So as a snapshot, the first core added value of our technology is the scale-up. We can mass produce billions of iPSCs for a much lower manufacturing cost. The second part is the quality of the cells, which is one of the highest standards in terms of genomic integrity data, cell viability, and stemness, and I'm going to dive into that a little later. And finally, what makes us a true biotech company is the efficacy. We are designing 3D, fully differentiated microtissues ready to graft, and that's our angle at developing cell therapies. So some key figures about the company. We started TreeFrog in Bordeaux, France in 2018. We are a team above 60 people now. We'll be over 100 people next year. And we just closed our Series B financing, including American investors last month. And I'm just mentioning that because that's going to help us tremendously expanding to two key markets for us, as we're already based in Europe. The first one is the US market with our Boston hub. So I'm the first US employee. Kevin, our co-founder, will move to Boston to lead the development of that hub. We'll be recruiting about 20 people by the end of next year, and we've been accepted to a lab space called Lab Central 238, sponsored by Astellas Pharma, so it's a terrific lab space in Kendall Square, so really excited about that. And the second space is also in Kobe, Japan, because it's an obvious, very dynamic ecosystem when it comes to iPSCs, so it's very important for us to be there. And the two key upcoming milestones for us is first being GMP compliant next year. Our technology right now is GMP-like, and I will dive into that a little later. And then obviously with the upcoming milestones to design a first-in-human trial by 2024 for our lead program being Parkinson's disease. So let's dive into the most interesting part of the presentation, our technology. The name of our technology is System. And so as you can see on the right part, we have a high throughput cell encapsulation device. And so what happens is we encapsulate between 3 to 5 stem cells in a capsule. And so the stem cells are going to self-organize, so to speak, as a cyst with the hollow in the middle, as you can see. Which is very important because that's what differentiates from aggregates, for example. With aggregates, you could have sometimes a necrotic core. That's not the case here, as you can see. And it's an epiblast-like structure with a monolayer of cells, so they also have equal access to nutrients, growth factors, and even differentiation factors. So the cells really love this 3D environment to grow, and if you compare that to 2D protocols, for example, there's no cell stacking in a flask. We also think it's a much better way to amplify your cells. And yeah, the picture on the left is essentially a 3D modelization of what's happening on the video on the right part, with the stem cells in the middle as an epiblast structure, a 3D environment, and envelop into their own matrix, and then into the capsule that is made of alginate. The alginate capsule also provides, protection against shear stress in the bioreactor. Also, it prevents from, the cysts to merge together, which will happen in a 3D amplification method with aggregates. So it's very protective. And very flexible technology. And that's probably the most important slide of the slide deck, so I'm going to talk a little bit more about this. But essentially, the entry point is you could take your stem cells and your matrix, and you run that through a alginate hydrogel based on microfluidics. And what you obtain at the lower left part of the graph is that seeding moment with 3 to 5 stem cells, as I was mentioning, self-organization as a cyst, And then once you reach about 80% of the inner volume of the capsule, that's our kind of confluence, so the cells won't amplify much after that. So essentially what we're doing is we are de-encapsulating, and the capsule is made of alginate, so it relies on the concentration of calcium in the media. So you just wash the media, get rid of the calcium, and it's pretty flexible. The capsule is going to dissolve in less than 60 seconds. And so you obtain your cyst without the capsule, and then with a cocktail of different enzymes, you just go back to cell suspension. And at TreeFrog, what we call passages is essentially, if you want to still amplify your cells, you just have to do another encapsulation run. So it's kind of like looping the left part of the graph here. And then the very interesting aspect is we can also differentiate our cells through the capsule as it is porous. And that's the most added value part of system. That's what makes us a biotech company. That's how we design our therapeutic pipeline. And so it's essentially the same principle. You seed your stem cells, they organize as a cyst, and once you have that cyst conformation, you run your differentiation protocols. And so you are designing 3D fully functional and matured microtissue that are ready to graft. And that's where we are very interested in co-development opportunities. We use this technological platform to develop our own pipeline, but we can also use system for partners willing to design a cell therapy together. And essentially, we're designing a unique biological object as an end product with unique properties. So we're talking, brand new target product profile and other properties with this end product. So that's we're very thrilled about this aspect of the technology. And that's as a snapshot. Once you have your cardiac cells, that's what we have on the left. It's beating cardiac cells within the capsule. Actually, I don't think the video is working, but you have the video showing you the dissolution of the capsule in less than 60 seconds, like I mentioned. And on the right part, it's the dissolution of this microtissue. If we wanted to, this 3D microtissue, we could dissolve it and go back to cell suspension mode if that's what we want to do in terms of injection method or delivery method, for example. Yeah, that's actually working now. So you can see the capsule is dissolving, and finally the microtissue go back to cell suspension. So it's very versatile and flexible technology, depending on what's the angle in terms of designing your end product. So again, the first part of the added value was the scale-up. We have a high throughput encapsulation device, and we want to be GMP compliant next year. So we're talking single-use bioreactor, fully automated and enclosed system. So that's a very, very big milestone for us next year. And what we've done, the latest publication we've done is we run 2 batches with the same cell line with 50 million cells as an input and 15 billion cells as an output in less than 7 days. So that gives us an amplification full of over 275 in a 10 liters bioreactor. So that's a very big milestone for us in terms of iPSC's expansion. And next milestone for us is to aim for 100 liters and then 1,000 for specific indications where you really need a lot of cells to produce. But it's not just the number of cells, it's also the quality, as we have over 98% of viability and very high pluripotency, so you can still differentiate those cells at the end. And that's the transition to the number 2, the quality of those cells. Here we run a comparative study between system and 2D protocols or aggregates. And as you can see on the left part of the graph, with system, you can grow much more cells than the 2D cell culture protocols with much less passages. So it's much less labor, much more automation. So we really think that's the right way to amplify your cells. And then in terms of genomic integrity, we have less oncogenic mutation than aggregates, for example. I can share much more data around that benchmark under NDA. If you're interested, I can share that with you. And that's not it. That's where we started, but we're also building a consortium, specifically around quality control. So it's made out of 4 people: Dr. Maxime Feuilleux, our co-founder and CSO at TreeFrog; Dr. Nathalie Lefort at Imagine Research Institute based in France; Dr. Shin Kawamata, director of RBI in Kobe, Japan; and finally Dr. Lawrence Dyron, based at Boston with the Harvard Stem Cell Institute. And last but not least, the efficacy. That's the picture of our lead program. We're making dopaminergic neurons, as I was mentioning. So on the left part is a 3D representation of that microtissue. It is fully differentiated and functional microtissue, dopaminergic neurons, not progenitors. And so what we've done in terms of in vivo proof of concept, we used the Emma Parkinsonian pre-graft model with the mice model, and we observed after 8 weeks after the transplantation a much faster recovery compared to 16 weeks after transplantation when you are using progenitors. And so, yeah, that's a fully motor function recovery that you can see on the website to see the videos. And that's our lead program. We are scaling up, so to speak, preclinical regulatory study on monkeys, monkeys being the best animal model for Parkinson's disease, to justify a first-in-human by 2024. That's our big milestones in terms of clinical trials. And that's our pipeline. Again, Parkinson's disease being an in-house program, most advanced in vivo proof of concept stage. We have a program based on cardiomyocyte as well. We use stem system as a platform to design several cell therapy based on different indications, and that's where we are very open to partnering. We just started opening the door of immuno-oncology applications, so that's a big door we just opened. We are doing a lot of due diligence with potential partners, and that's why the MESA is an amazing conference to attend on that aspect. And we have a bunch of undisclosed programs, but at a discovery stage so far. And that's the one slide I have for today. As part, being part of the BD team, our job is to co-develop cell therapies with partners from really the bench top to the market. And so what we can provide is adapt. If you have a 2D protocols, we can adapt that to 3D and use system with your cell line, and together co-develop a product on a specific indication that we just lock in together. We also can provide what we call early scale-up, and by that we mean manufacturing of non-clinical phases, non-clinical lot, and up to the phase 1, or at least early clinical phases. We also want to supervise everything from A to Z, including the delivery. So we are developing our own medical device for this local delivery of the 3D microtissue that I just shown you. And finally, we want to optimize manufacturing as well. So when we have a preferred partner and we are entering later clinical phases and hopefully post-approval and then commercialization, we can tech transfer system to your site with the supervision of TreeFrog, obviously. So that's it from me. Thank you for your attention. The motto of the company is cell therapy for all, because we really want to drastically reduce the cost of manufacturing for cell therapy. And hopefully one day every patient will be able to afford cell therapy. That's the goal here. Thank you for your attention, and I can take a couple questions. I have 1.5 minutes. Thank you very much. Yes. Thank you. Yeah, that's a terrific question. So actually, it's what we've been optimizing around the capsule in terms of size and how can we influence, I guess, the amplification fold of those cells within the capsule. So we've been playing around a lot with the capsule. We have so far 2 different, I'll say, size of capsules. For example, when we grow cardiomyocytes, we use the bigger size than the one for dopaminergic neurons, for example. We're playing a lot around with the kind of polymer that is made of the capsule. So far, it's alginate, but we can also anticipate different polymers that will be also approved by the FDA for the US market. It's very important to have the right polymer. We can also use this capsule as a cell delivery method, if you will. We can also play around the level of, how porous exactly is the capsule. We can also play around that. So it's a very flexible tool and versatile, like I was mentioning. I'm happy to continue the discussion and get into details under NDA, etc. But yeah, that's how we've been playing around with the capsule. I think that's it. But I'll be at the back of the room if you want to follow up with some questions. Thank you very much for your attention.